as well as the today's most respectable speaker, Dr. Benjamin A. Horwitz from Israel, who readily agreed soon when I informed him about our IPS Platinum Jubilee lecture series. So it is indeed a great opportunity and great privilege to welcome him. I think all the trichoderma research workers and the students, and they are all very familiar with his name. But still then we need his introduction. So I would request further that after we can introduce our speaker, Dr. Benjamin A. Horwitz, professor from Technion University, Israel. He may be introduced by Dr. Aruf Mukherjee. So Dr. Aruf Mukherjee, kindly introduce Dr. Benjamin.
Good morning, madam. Thank you very much. I am privileged to introduce Professor Benjamin A. Horwitz, or Benny as he is known popularly among his students and others. He was born in Boston, USA, 1955, but interestingly, leaving all the comforts of USA behind, he joined as a postdoctoral fellow in the Institute of Carnegie Institution as well as Syracuse University. And he did his PhD from Weizmann Institute of Science in 1984. And 1987, he took charge as a faculty of Israel Institute of Technology at Technion, Haifa. And he set a lab in the cell signaling pathways of filamentous fungi, in particular their relevance to fungal plant interaction. I had the fortune to work with him for 1 year from 2006 to 2007. And I personal experience is that I have seen so many persons, but such a nice personality, who is excellent human being as well as excellent academician. It's very rare qualities he's having. Always with a smiling face, and he is at service for all his students as well as any other who are near and dear ones for 24/7. And I have seen his extraordinary publication in plant physiology, PNS, plant cell, and what not. All high-rated plant science journals he had published. And I have seen him to work for 16 hours to 17, even 18 hours I have seen continuously he works. He sits on the chair and he works, and he's always ready to help everybody. And I feel that the tremendous contribution he has made in plant pathogen interaction, or you can say maize cochlebolus interaction as well as trichoderma genetics, is I cannot say that it is praiseworthy because I am not about I myself is not at that standard that I will evaluate him, but I have a huge regards for him as a person as well as an academician. And today he is going to enlighten us with his lecture on maize cochlebolus heteroscopus interaction. And Benny, now the stage is yours. We are eagerly waiting to listen to you. Thank you, madam, for giving me the opportunity to introduce such a great man.
Thank you so much for the kind introduction, Arup. And well, I'll have to live up to some expectations. And thanks to Pratibha and to the Society for the chance to talk in this series. I will just share screen now. Okay. So this is a story about a plant pathogen and our attempts to understand cell signaling in this pathogen. And I'll really tell you the story from the beginning. So the parts that are published already, I'll go faster, and the more recent things, I'll go into a little more detail, and then we can discuss afterwards. So as Arup said, I'm at the Technion in Haifa, and I'll be talking about the maize pathogen Cochleobolus heterostrophus. And in this photo, which was from one of my visits to the lab of Gillian Turgeon at Cornell, which is a lab which uses Cochleobolus system as a model pathogen, even though in the field, it's not so much of a problem because farmers don't plant maize, it's sensitive to the T toxin made by Cochleobolus. But on these leaves, you can see lesions formed where spores have fallen on the leaf and germinated. And it varies a bit depending on the maize cultivar. Here they look light on the inside on this particular line, but they can be darker on the inside or even larger and more necrotic. So it varies with which particular host they're on. The pathogen comes in two mating types, MAT11 and MAT12, and it's nice that these can be crossed in the lab so that one can do classical genetics if you want to. It's not maybe the easiest genetic system to work with, but you can do crosses and analyze progeny from ascospores. And on the left is MAT11, which is actually a strain that doesn't make the toxin, and on the right, a strain that does make the toxin. And the lesions caused by these two strains are fairly similar. So they're almost perfectly isogenic backgrounds, other than toxin production in the right-hand side and the mating types themselves. And because this maize strain is not sensitive to the toxin, the symptoms are the same. If the toxin plus strain were sprayed on a sensitive maize, the leaves would be just bleaching and falling over, and you get these streaks on the leaf where the toxin is doing its work, killing the leaf. But I'll be talking not about the toxin, but about signaling, how the fungal cell recognizes that it's on the host surface, leaf surface, responds to it, and reprograms its development and gene expression accordingly. So the story goes back to the 90s when the first signaling related genes were identified in filamentous fungi. And one would hope that the phenotype, if you knock out a signaling gene, the fungus should grow normally, but maybe it won't respond to signals. So it should just look normal, viable, but maybe not be virulent or not be able to mate or not be able to develop. In reality, the mutants that were isolated in this really past 30 years of work using this approach by many other labs and ourselves, is that the mutants tend to be pleiotropic. They have many different defects, some which may be related to response to the host or to developmental pathways, light, and so forth, and others that seem to be secondary developmental phenotypes, that when cells lack these genes, they can't do things that the wild type can do. So, but nevertheless, a lot was learned. And the first gene, if you look at signaling, so signaling comes from outside. If it's a chemical signal, it's detected by a receptor, often a membrane localized one, not necessarily, could be intracellular as well. Receptor passes the signal to a signaling element like this heterotrimeric G protein. And when looking for the first gene to knock out and to try to look at signaling pathways, we first chose the alpha subunit of heterotrimeric G proteins because it's such a conserved gene. And this was before the first filamentous fungal sequences. And when we knocked out, deleted the gene for one of the three G alpha subunits in the genome of Heterostrophus, which at the time we didn't even know how many are there, it was again before sequencing. We saw an interesting phenotype. And if you look at the germinating spores, these are about 50 micron long, sort of banana shaped, multicellular spores. There are cross walls here that you can't see so well, but they're there. And this spore germinates often from the two ends. So it used to be, and is still called sometimes bipolaris, matus, instead of C. heterostrophus. So that you see germ tubes coming out. The wild type on a hard surface will grow a bit and then form a small aprosorium-like structure. It's not the large aprosorium that you might see in Magnaportha griseum, in rice blast, for example, where the aprosorium is really almost as large as the spore and generates huge turgor pressure. Here it doesn't. They're small, little pillow-like structures. And from there, the fungus will invade the leaf mainly by enzymatic activity and not by building up turgor pressure. So the wild type germinates in a meandering path like this and often forms small archosaria. On the other hand, the phenotype we saw when G alpha subunit was knocked out was that the germ tubes would grow straight as if they can't see the surface. And even 20 some years later, we still don't know the cell biology that underlies this effect. We don't know why when these hyphae lack G alpha, they grow straight rather than in a meandering path, and then they don't form aprosoria. Nevertheless, these mutants can make symptoms on maize leaves, maybe a little less than wild type, but they still cause symptoms. The next gene that we looked at in the pathway, and I will just try to move this bar out of the way. Let's see. So it's not covering the title of the slides. So the next gene that we looked at was one of the 3 MAP kinase genes encoded in the genome. And by then we were able to use the genome sequence that was just in process. So MAP kinase cascade is a series of 3 protein kinases where the signal comes in, results in phosphorylation of the upstream MAP kinase, or MAPKK, or MEK. So we see the phosphorylation and eventually dual phosphorylation on the MAP kinase, of which there are 3 in the genome. And when Sophie Lev, who was a PhD student in the lab, knocked out the first one of those 3 that we looked at, and this one, we named it at the time CHK1, or Cochleobolus heterostrophus kinase 1, but CHK1 is the name of other cell cycle related kinase, and in rice blast, the ortholog of CHK1 is called pathogenicity MAP kinase, or PMK1. So we now try to remember to call this PMK1. So when Sophie knocked out PMK1, she got, again, really a pleiotropic phenotype. And if you just look even at the colony stage, so at the right here is wild type in the light and in the dark. And in the light, the mycelia tend to branch a lot, make conidia. In the dark, we get this fluffy aerial growth without conidia. That's the wild type. On the left, you see the mutant, and the mutant hyphae tend to stick to each other. They become easily wettable. And in the dark, you see that this white colony with a ring of internally hyphae that are just falling together and sticking, and that shows up in the scanning EM where you see some extracellular material deposited on the hyphae, and in the wild type that's not there. And again, the mutant has a difficulty in sensing that it's growing on a hard surface, and PMK minus mutants don't sporulate, but you can grow them on a glass slide, allow them to grow in liquid medium, and the wild type will eventually adhere to the substrate. And if you look closely in the higher magnification, it will make these small aprosoria, just like germlings do, whereas the mutant will even run in circles because it, again, is missing the point that it's on a hard surface. It doesn't make aprosoria, and it will often form these wavy, very wavy growth, unlike the G protein mutant that grows straight. Now, if you try to cross things, the wild type, here's the typical wild type cross. You can do this in the lab with a senescent corn leaf, maize leaf, and you see the pseudothesia or parathesia, except that the outside is formed from vegetative hyphae, so we call them parathesia, with the asci and ascospores inside. And these are heavily melanized and appear on the surface of the leaf. Now, if we try to cross two mutants, which are both lacking our MAP kinase, we don't see any pseudothecia at all. Whereas if one of the parents strains is wild type, then we still get perithecia, pseudothecia, but the mutant is female sterile in the sense that the tissue of the pseudothecium has to come from the wild type parent. If the parent is lacking the MAPK, the hyphae don't differentiate properly, and all of the pseudothecia are formed from the wild type parent. And here, finally, inoculating, it's a rather extreme laboratory inoculation where at 2 points on the leaf, Sophie put a droplet inoculation with about 100 spores in it. And a week later, the leaf has become completely necrotic. But inoculation with the mutants causes almost nothing, just very weak symptoms on the leaf surface. So here we have a mutant that really grows quite nicely, grows like wild type, but is practically avirulent. The next MAP kinase that we looked at among the three, and I'll only be talking really about 2 out of the 3, is the stress-activated MAPK, which is an ortholog of mammalian P38 and yeast HOG1. And we call it HOG1, Cochleobolus heterotrophus HOG1. So when Aidic Baria, who was a master's student in the lab at the time, knocked out HOG1, what he saw was a colony that grows almost like wild type, a little bit slower. In contrast to PMK1, we see normal profuse sporulation, formation of conidia on the colony surface, grows a little bit slower. And one thing that we noticed later on while growing cultures of this mutant is that if you grow too long in culture, and especially in shake culture, the mycelia tend to go into cell death. So it'll stop growing, sit there. When you shake it, you see, you'll see these spherical, typical spherical colonies in liquid culture, but at the colony borders, the cells begin to die. And this is evident here in staining with trypan blue. So here's a part of the colony edge where most of the hyphae have died, whereas in the wild type, once in a while, you'll see a few cells that are no longer viable, like this compartment here, but for the most part, the colony is viable and growing. Sporulation, as I said, on the y-axis, millions of spores per plate. Wild type, HOG1 mutant, even within the variability, is sporulating a little more than wild type. And the third MAPK, MPS1, or cell integrity related MAP kinase, that one, like PMK1, is deficient in sporulation. So HOG1 seems to be involved in helping the cell to survive stresses, and this will become more important, more apparent later on. So one more property of HOG1, if we look at a couple of genes that are conserved and known to be downstream of HOG1 in yeast. So one of these is a monosaccharide transporter and a transaldolase. Response to osmotic stress involves changes in sugar transport because the cell is trying to accumulate osmolates. And if you look at expression, this is by old-fashioned before qPCR, RT-PCR. So MST1 is strongly induced in PMK1 mutant, even in the cell integrity MAP kinase in the wild type, but not in delta HOG1 by exposure to molar concentrations of KCl. And the fungus can survive that strong osmotic stress, but not if it's lacking HOG1. Similarly for TAL1, maybe not quite as clear some of the picture, but in the wild type, we see strong induction of TAL1, and in the mutant, nothing at all. So both for expression and for osmolite-dependent expression, HOG1 is required. Now, going fast forward to 2014, 2016, Summer Shalaby, a PhD student in the lab, found that exposure of Cochleobolus to predicted plant defense compounds like ferulic acid or caffeic acid, and in a minute I'll say what's different about these two, but exposure to some phenolic acids will, to make a long story short, send the fungal pathogen cells into programmed cell death. And here's what that looks like. So here's the DMSO control. DMSO is just used as the solvent for phenolic acid. Exposure to ferulic acid. If you look at the aprosuria, just like I showed before, they look normal, but exposure of the colony to ferulic acid will permeabilize the cells, and you can see the aprosuria, this time stained with trypan blue. 20 minutes after the start of exposure to ferulic acid, the cells begin to die, and you see these, unfortunately it's a black and white photo, but you see these accumulating and staining with trypan blue. At 4 millimolar ferulic acid or 2 millimolar, which are concentrations that, it's interesting, no one knows exactly what the concentration is at the location that the pathogen sees as it's going into the leaf, but millimolar concentrations are physiologically realistic. On the other hand, caffeic acid, which differs from ferulic only by this one side chain, instead of the methyl ester here, the methoxy group, is caffeic acid has adjacent OH groups. And the difference here really is that when the cell tries to metabolize this phenolic, the fungus can cut this bond here and send the phenolic into the alpha ketoadipate pathway, and from there even use it as an energy source, whereas ferulic acid is much more difficult to hydrolyze because it doesn't have the adjacent OHs that caffeic acid does. And caffeic acid causes no cell death at all. And the type of cell death, and here I just maybe digress for a minute, we have to remember I'll be talking more about the MAP kinase HOG1. But if we look at cells that have been fixed and permeabilized and stained for apoptosis. Now I'm careful not to call this cell death apoptosis because in the sense of mammalian apoptosis, this is a rapid cell death. It shares a lot of properties with apoptosis, but we would rather call it regulated cell death or rapid regulated cell death since we don't know exactly how it happens. But a kit which is designed to follow the nicks that occur in DNA during mammalian apoptosis. And the way this works is DNA that's been cut, terminal transferase can come in and attach a modified base to the end, to this terminal 5' end of the DNA that's been cut. And then the stained preparation is treated with an antibody that recognizes the altered nucleotide. And then wherever DNA has been cut, you see a bright spot, which is a positive nucleus. Now it's important to realize that the spores here don't take up the reagents. So although there is cell death in the spores, as we'll see in a minute, using a different assay, this tunnel assay, TUNEL or apoptosis assay, does pick up positive cells in the mycelia that are overlying, growing over the spores in these photos. So some of these lines that are out of focus in the bright field, but the confocal plane is picking up the fluorescence. So in the DMSO control, we see no positive nuclei, and treatment with ferulic acid at 10 minutes already, we see positive cells. And by 20 minutes, we see many positive cells, quite comparable to what happens when these same preparations have been treated with DNAs to nick the DNA. So at least this process shares with apoptosis. It shares the fact that the DNA has been nicked. Now, again, exactly how that happens, we don't know. Another assay which shows that this process is similar to programmed cell death is to take a line that expresses histone H1 GFP. And we can see in control nuclei, I didn't say so, but the conidia are multinucleate. So we see many fluorescent nuclei in the spores, and in the cells also, the nuclei are fairly free to move between the cell compartments of the hyphae here. And we see fluorescent nuclei when the germlings are treated with ferulic acid. Within 15 minutes, we can already see that histone H1 GFP is dispersing from the nuclei. And at 4 millimolar, this is even more striking. So after a few minutes, histone H3 has started to disperse, whereas treatment with caffeic acid right here, no effect at all. And now finally back to HOG1. So what Summer did in this slide is to treat spores at time 0 with ferulic acid for 20, 40, or 60 minutes, wash away the phenolic acid, plate, and a day later, look at survival, count colonies. So really very simple assay. And we see that over the hour of exposure to ferulic acid, the wild type loses viability. So ferulic acid is toxic even for wild type cells, but lack of the MAP kinase HOG1 results in much more severe cell death. And when Summer looked at phosphorylation of HOG1, to his surprise, it turned out that rather than being phosphorylated by stress, as one might expect, all MAP kinases in mammalian cells, there's no basal level even. So all MAP kinases, you give the signal, and then one sees strong phosphorylation, dual phosphorylation, as I said before, on a conserved motif that TXY, where X is can be, in this case, a glycine, TGY in HOG1, or glutamic acid, TEY in PMK1, or the ERK family MAP kinases. So, but when Summer looked at phosphorylation in response to exposure to ferulic acid, to his surprise, rather than seeing increased phosphorylation, he saw a decrease. So HOG1, by the stress-like stimulus of FA, which causes cell death in these cells, rather than increasing phosphorylation of HOG1, it caused a dephosphorylation, quite striking dephosphorylation. Looking at the other two MAP kinases, the cell integrity MAPK, MPS1, showed increased phosphorylation, not such a strong response, but maybe 2-4-fold increase. And PMK1, CHK1 showed at first a dephosphorylation and then a return to the steady state level. And these are this antibody here recognizes the total amount of all three MAP kinases as a loading control or a total MAP kinase signal. So I forgot to mention this is a Western blot. So the antibody here is specific to dual phosphorylated HOG1, and here to dual phosphorylated MPS1 or is CHK1, PMK1. So HOG1, stress-sensitive MAPK, is dephosphorylated by exposure to ferulic acid. And in Cochleobolus heterostrophus, as in many other fungi, there's a significant steady state level of the MAP kinase before exposure to any stress stimulus. Of course, we don't know. Maybe growth in culture is already perceived as a stress. And if this pathogen was growing on its host leaf, maybe the our so-called background level would be lower. But at least as a biochemical experiment, what we see is a steady state background level, and exposure to ferulic acid dephosphorylates. Now, can we raise the phosphorylation level of HOG1? The answer is yes, and I'll show that a little later. But our approach here, and what Summer began to ask, is what would happen if we couldn't dephosphorylate this MAP kinase? So could dephosphorylation be a signal rather than phosphorylation? We know that HOG1 is important. When it's missing, the cells are less viable when exposed to ferulic acid. So could it be that dephosphorylation is also a signal? And that's what I'll talk about in the next few slides. And to answer this question, we started to look for the protein phosphatase genes of Cochleobolus heterostrophus. So we asked, can we knock out the phosphatase? And Summer had finished his PhD, and the next people to work on this project were Rina Zuchman, PhD student in the lab, and Ronnie Koren, who is my lab manager. And what Rina and Ronnie did was to look, luckily there was information already in the literature about protein kinases in filamentous fungi, in Neurospora, Aspergillus, these genetic model systems. And this is from a paper from the group of Gustavo Goldman in Sao Paulo, Brazil. And the ones shown highlighted in light blue are the first four that we chose to make knockouts of. And the first thing, of course, when we found out how many there are, is we said, well, one can make knockouts in Cochleobolus, but there's some 24 here in Aspergillus, and assuming that it would be similar in Cochleobolus and another astomycete, that would really be a lot of protein phosphatases to try to knock out. So we looked in the literature from yeast, which ones might be involved in dephosphorylation of HOG1. Excuse me, I just need to connect my power supply. So there we are. That was making this screen a little less bright. So PTP1 and PTP2 are tyrosine protein kinases that are involved from the yeast literature in dephosphorylation of activated HOG1 in yeast. Aspergillus fumigatus, PTCB, from this paper is involved in dephosphorylation of HOG1 in the human pathogen, Aspergillus fumigatus. And CDC14 we added on because it's an intriguing protein kinase because it's a dual specificity kinase. So here's Cochleobolus CDC14, fungal homologs, and mouse as an outgroup here, C. elegans. So we see it's related to its fungal orthologs and it has the correct domain. PTP1 and PTP2. So these are the yeast proteins. PTP1, PTP2, and PTP3. In yeast, there are two genes because of the yeast genome duplication, but our PTP2 groups with the yeast proteins. There's mouse as an outgroup, and it has the correct domain. And PTCB, which belongs to the, I'm sure you can't read this, but it's a PP protein phosphatase 2C superfamily domain. So PTCB belongs to the PP2C superfamily. And if one knocks out these genes, and Rina made knockouts for all these 4 genes, and then we could start to look at the phenotypes. So the first thing that we were looking at, just to remind, I'll jump back to the scheme here. So we were interested in seeing what happens if we can knock out a phosphatase and block dephosphorylation of HOG1. So that would seem to be bewildering with so many phosphatases. But as I said, we were guided by the literature and made knockouts of 4 genes. And when Rena looked at dephosphorylation in response to exposure to ferulic acid in mutants in those 4 phosphatase genes, here is PTP1 and PTP2 compared to wild type. In wild type, we see dephosphorylation. These are 3 replicates, so sometimes you don't see it so clearly, but quantitatively, there's the total. So total goes up a bit, this goes down. Quantitatively, if we look at wild type, dephosphorylation responds to exposure to FA. Looking at PTP1 and PTP2, the tyrosine phosphatases, we see dephosphorylation. And here's the average PTP1 and PTP2. Mutant in PTP2 might show a little bit of loss of the phenotype, the wild type behavior, but it's not significant. So sometimes we see a little bit less of a decrease. But what's really dramatic is that CDC14 and PTCB mutants
नहीं एक लिए हैं
have
जो ऊपर लटकाई है वही लिया हूं मैं
have almost completely lost the ability to dephosphorylate HOG1. So we might say maybe this whole story is an artifact and what's happening with phosphorylation, and it's a filamentous fungus, and we're using an antibody. Our anti-P38 is for mammalian P38. It works beautifully in Western blots of fungi, as I showed in an earlier slide. And there's the total HOG1, again, a mammalian one. And we've now made antibody to the cochleobolus sequence. But if we look at phosphorylation in response to 1 molar sorbitol, and this is what HOG1 was meant to do. I mean, HOG1 is the high osmolarity glycerol pathway. That's what HOG stands for in yeast. So if we expose to sorbitol, we see dramatic phosphorylation in response to high osmolarity. So HOG1 is working, but when we lack PTCB, we don't see the dephosphorylation response, whereas rapid phosphorylation in response to sorbitol is normal. So here's the fold phosphorylation. The bottom of this graph should be 1. That's a mistake. One always finds these things while lecturing. So plus sorbitol divided by minus sorbitol should start at 1. And we see like a 7 or 8 or more fold phosphorylation in response to sorbitol, but somewhat less in response to in response to sorbitol in CDC14. But what's really nice is the comparison between the response to FA and the response to sorbitol, lacking the phosphatase PTCB. This one behaves just like, this mutant behaves just like wild type. So we need PTCB for dephosphorylation. We also need CDC14. So how can it be 2 unrelated genes, knockouts, we see a similar phenotype? And we're working now on trying to think of sequential dephosphorylation models and why several phosphatases might be needed. Interestingly, we started with PTP1 and PTP2 from yeast, and these are normal or near normal dephosphorylation. So our filamentous fungus does not behave like yeast, at least in response to ferulic acid. And one thing that Rena has done just recently is to construct a HOG1 protein fused to GFP. So in the spinning disc confocal, here's here hyphae expressing HOG1 GFP, and you can see fluorescence in the cytoplasm and in the nucleus. And these are fairly new data, so they're very preliminary. Still need to do more controls and look at mutants and so forth. But what we can see with sorbitol, here's CM complete medium. HOG1 is distributed in the cytoplasm, also some in the nucleus. Exposure to DMSO doesn't do very much, some in the nuclei, some in the cytoplasm. Exposure to sorbitol, CM, complete medium sorbitol 1 molar, for 5 minutes, we already see HOG1 GFP in the nuclei. So this left side of the slide is known. This has been done in other systems. Beautiful pictures from rice blast, where you see the same thing. On the other hand, exposure to ferulic acid does not send HOG1 into the nucleus, which might have been expected because it's not phosphorylated, but until we see it, we don't know. Instead, we see accumulation of HOG1 GFP in cytoplasmic foci, which we still don't know what they are. So these might be fragmented nuclei because we know that exposure to FA causes histone H1 GFP to diffuse or go out of nuclei. Another thing that's crossed our mind is that these might be stress granules, that maybe phosphorylated HOG1 is being sequestered in stress granules. We really don't know, and this is something that we would like to work on. So I'll close by describing the phenotypes of this third MAPK, and then maybe just a quick mention of some other aspects of signaling that we've looked at. And then after that, we can open for discussion. So looking at the phenotypes of the phosphatase mutants. So here's HOG1. And Rena saw, as I did some years ago, that HOG1 grows a little slower, but sporulates profusely. Phosphatase PTCB, on the other hand, shows defective growth. And interestingly, appearance of sectors here, which we don't know what they are. Some phosphatase mutants in other fungi have also show suppression or sectoring. CDC14 also shows somewhat defective growth. The tyrosine phosphatase mutants, on the other hand, grow beautifully, look just like wild type. And this is after 2 weeks. So part of what's happened here is that HOG1 not only grows a little slower, but the colony edge is becoming less viable. So the growth defect is really more extreme than it would be after, say, 3 or 4 days. Looking at virulence in these mutants, so some of them don't sporulate, so which makes it a bit of a challenge to inoculate them. But what Rena did was to cut little mycelial blocks from the colony edges, and we stick them to the leaves, and 24 hours after, remove the tapes, and then incubate them normally. And 4 days after inoculation, the wild type shows large lesions. HOG1, this should say HOG1. Delta HOG1 shows lesions that are similar to wild type. On the average, sometimes a little bit smaller, and that is significant. Here's lesion area. So Delta HOG1 is a little bit, but it still causes lesions. On the other hand, PTCB has drastically lost virulence. There's PTCB. CDC14, although it's very slow, slow growth and doesn't look very normal as a colony, but it can still cause lesions, but much less than wild type. And the tyrosine kinase, the tyrosine phosphatase mutants, PTP1 and PTP2, look normal, just like wild type. So yeah, looking at the schedule, I guess I should be stopping soon. So I'll just mention that we've been looking at dedicated fungal transcription factors that are involved in sensing stimuli. And this is published, so I will stop after this slide and open for discussion because we're getting to my 7:45. So Sophie looked at a transcription factor which is sensitive to redox state. And this one we call CHAP1 or CHAP1. And when Sophie knocked out this transcription factor, she got a line that's very sensitive to oxidative stress. And here, if we look at the GFP fusion of the transcription factor, it is nuclear localized and controls genes that are involved in the response to oxidative stress. And there's a line of work in the lab that we're looking at this transcription factor and the genes that are under its control. And it's also involved in survival under stress. So the people involved in the story that I told, which is signal transduction, but mainly the MAP kinase HOG1 and the phosphatases that dephosphorylate activated HOG1, are Rena Zuchman, who's current PhD student in the lab, and Ronnie. My group is quite small these days because of the pandemic or other reasons. There's nobody interviewing, so no new students have joined the lab recently. So we're a little group, but working mainly on this phosphatase question. Summer, who was a PhD student and then went on postdoc to New York City, and Orit, or Orita for short, who was our lab manager at the time. Now Roni. So these people I've mentioned, and the other past project in the lab, which we're still trying to keep going, is on trichoderma. And Prasun Mukherjee, when he was visiting the lab, and this is already over a decade ago, really kept my interest in trichoderma going. And these people listed here in green were a trichoderma subgroup. So Ariela, Renat, Jamila, Fernando, and Carmen, who was visiting from Mexico, worked on trichoderma, RNA-seq, and everything genome-wide, which I didn't talk about today. Technion Genome Center, the Turgeon Lab at Cornell, who are longtime collaborators on Copiobulus, support from Israel Science Foundation, from the US-Israel Binational Agricultural Research and Development Fund, BARD, and JGI, who did the sequence, without which none of this would have been possible. And this is just to remind me to thank once again Professor Pratibha Sharma, president of IPS, for inviting me to talk in this series. And this picture, here's Prasun and Arup, who just introduced me, and Prasun's thesis advisor at a meeting that Pratibha actually was the main organizer of and sponsored his IPS meeting in 2016 on trichoderma. So yeah, thanks again. And I guess I'm pretty much on time, a little late, and I'll take any questions or discussion.
Thank you, sir. Thank you for excellent talk. Now we are open for discussion, please.
Yeah, so one point which I didn't get to, which could be something to discuss, because it's something that we don't really understand. This is a model. So I spent quite a few slides talking about RCD, or regulated cell death. And this is a slide which actually is taken from a review on apoptosis, or cell death in animal cells and adaptive stress response. So what they're saying in this review is that the cell first tries to survive, and then it goes into cell death, which is really something you would might expect. And when some of these signaling genes are lost, in this case, it's the transcription factor that I just briefly mentioned at the end. So in the mutant, the adaptive response doesn't work so well, and then cell death kicks in earlier, or at a lower level of stress. And we got to this by looking at gene expression in the mutant. So in the mutant, at concentrations of ferulic acid, which we've seen acts as a stress, so concentrations that the wild type is not happy with but survives well, the mutant already seems to be going to cell death according to gene expression. So the pathogen goes into cell death, and this has been shown by others. Surprisingly, on the leaf, some necrotrophic pathogens go into cell death, but they survive. They start to grow again. Okay, so they come out of it, but the mutant might not. So lacking signaling genes, the mutant can't respond properly to stress. So that's a point that's, of course, that we would like to study. We don't know whether this model is correct or not.
Thank you, sir. You gave a gist of your talk in the last slide. Thank you.
So here, Dr. Prashant.
Yes, sir. Please, if you have any query, please ask.
Yeah. Yes. Thank you, Dr. Benjamin. Myself, Dr. Robin Gogoi, Secretary, Indian Phytopathological Society. Dr. Benjamin.
Yes.
Yeah. So
Yeah.
here, so whether this, how many transcription factors quantified in maize versus Cochleobolus heteroscopus this interaction system, whether quantified or still working?
I don't know because about maize because we don't work on maize. Just an offhand guess, but the genome is there, so you could look for it. And somebody may have compiled a list. In tomato, I think there's a list, but there are certainly more in the plant than in the fungus. In the fungus, there should be tens, and in Neurospora, the transcription factors have been there's a list, and most have been knocked out. In Cochleobolus, we don't know, so I don't know the answer to your question, but we have the genome, and there are tens. One could go and knock them out one by one. That's possible, and it's one thing to do. Another would be to knock out receptors, but yeah, so I don't have a full answer, but the genomes are there, so.
Yes, thank you.
So I see 3 chats. Let me see if there are, no questions.
Any questions?
Yeah, Dr. Horwitz, I am Anil Kotasthane from Raipur, and I would be interested in why we see a lot of brown spot that is again caused by Helminthosporium in rice when there is a lot of drought or water stress condition. So what kind of possible signaling activity might be going on, which actually is responsible to sense the drought and have an elaborate incidence of the disease in the field?
Okay, so yeah, so these plants, of course, are greenhouse plants, and they're under the most perfect conditions that could be, and they look very nice. We haven't looked at drought stress, but as you say, in the field, you probably see it. And one thing that we have noticed, one stress that we have looked at is lack of iron. Now, if you put Cochleobolus on leaves that lack iron, they may become more or less sensitive to the leaves become more or less sensitive to the pathogen, depending on the iron concentration. Another thing we've noticed, and this we have published, is that if you look at a leaf that's a little bit senescent, starting to turn yellow, becomes much more sensitive. So they're not under drought, but we looked at cut leaves. So detached leaves, even they have plenty of moisture, but they're cut, which is, they start to senesce, and then they're very sensitive. So something is similar is probably happening under drought stress, that the physiological state of the host. And the reason we did this was to look at wild type and mutant. And it turns out that wild type will infect plants that are in good shape, but the mutant, G alpha mutant, can infect better plants that are under, or leaves that are under stress. So again, we've never tried it under drought, which would be a good experiment to try. But that's probably the explanation. It's that when the host can mount a weaker immune response, then it becomes more sensitive.
Yeah. Another thing which we observe that the plants, they have increased levels of proline content there. Then is it? Yeah.
Increased proline. Yeah, I don't know offhand about the plant stress response. Yeah, I can't.
Okay. Thank you.
I think Dr. Arup wants to ask a question.
Yeah. Benny,
Go ahead. Yeah.
Benny,
Yeah.
just told that at the senescence stage, the plants become more susceptible, right?
To what?
To cochleovirus.
Are more
At senescence
susceptible
stage,
as a senescence.
at
Yeah.
senescence
Yeah.
stage, the leaves become more susceptible to your cochleovirus, right?
That's right.
Okay.
But we
In
haven't
that
looked at
case,
a
if we, yeah, if in that case, if we transform the plants with stay green gene, will the susceptibility will decrease? Is there any possibility?
We don't know, and of course, but that's a great experiment to try. So, are there mutants in the stay green? In which plant? Did
Maize.
expression?
I'm
So you've
talking about
expressed-
rice and maize.
Rice and maize. So they're transgenic overexpressors in rice and maize. Yeah, that would really be nice.
Yeah.
Try. And yeah, I mean, you can do it. Just spray them with Cochleobolus heterostrophus. So yeah,
Okay.
you have
Thank you very much.
Thorium biflorus. You have it in the field. So yeah, that would be a great experiment to try. I don't think that anybody has done it, but it would be worth looking whether anybody has looked at senescent plants. It might not be completely new, but it would be a great experiment to try.
Thank you.
Dr. Horwitz, yeah, again, Anil here.
Yeah.
Is the infection process in case of cochleobolus, it is invasive or it is through natural openings? And how the Epilosaria, it contributes towards the process of infection?
Well, we've seen hyphae going in through stomata, but also at locations where there are no stomata. So it's interesting that MAPK mutant, it actually accumulates over stomata, and it likes, so to speak, to go in through stomata, whereas wild type can go into the leaf wherever it wants. So there's both routes of entry.
Yeah. So will like exactly in case of Magnoporthecariaceae, where the melanization is there, and if you are interfering with the process of melanization, then altogether the infection process is eliminated in case of Magnoporthecariaceae. Will it happen in this case also if you?
Yeah, no, it does. It doesn't happen because Cochleobolus apparently uses enzymes to, if it penetrates directly. And in the Turgeon lab, they've made the knockout for melanin. And I showed a picture with the cross with an albino strain.
Yes.
So knockouts for melanin are pathogenic and under laboratory conditions. In the field, it will be different because it's exposed to ultraviolet, but in the lab, they're just like wild type.
And secondly, we have also observed that the pathogen or the spore, it produces a lot of Epilosaria from many cells. It may be from both the ends, but whenever the germ tube is being insured, it produces branching, and there it produces a lot of Epilosaria in several branches. So why it happens? So is it so it increases the efficiency of the pathogen to infect or something else?
So you said when it's damaged, when the germ tube is damaged?
It produces a lot of Epilosaria there.
Okay, that's interesting. Yeah, that's interesting. And I don't know why. My only comment comes from the trichoderma system, where Alfredo Herrera's lab in Mexico showed that if you cut a colony, it'll conidiate profusely. So damage acts as a stimulus, probably as an oxidative stress. So it's triggering this response. Why it makes more aperture, no idea. Evolutionarily, it's trying to penetrate that.
Okay.
Makes sense, but yeah, we've never seen that or tried to look at it. But,
Okay.
Yeah, I just have one question.
yeah.
Hi,
good to see you.
Yeah, good morning, Benny. To the distinguished audience, I had the privilege of working with Professor Benny for my postdoctoral program as part of the DBT project way back in 2007. And my question to Professor Benny is, often the plant response, there will be an overlapping response in PTA, that early response, and the response which is triggered after recognition of the pathogen effector by the R protein. So the responses which you have studied in case of cochleobolus to the maize signals are of early response. Is there any study where they have mutated this effector genes so that they are not recognized by the R genes, and then the response is specific only to the earlier response, that is the PTA or basal resistance of maize has been studied or not? Do you have any idea about that?
Yeah, that would be very interesting. Before I answer, I just like to say, yeah, I hadn't mentioned that that you had been in my lab, Dr. Shanmugam, and I didn't get to this chapter, but yeah, it's great to see you. And of course, you were involved in the work. That's about 12 years ago that you were in my lab, and this work was published in no less than Cellular Microbiology. So Dr. Shanmugam is first author, and this was a project where we identified ferulic acid as the component in plant extracts that caused the transcription factor that I mentioned to accumulate in nuclei. And now I'll answer your question, which will take me less time. In Magnaportha and rice blast, there's a lot of work on the effectors. In cochleobolus, we don't know anything about the effectors, and the genome is there, and it's just open for somebody to do that, to go and find the effectors and make mutants and knock them out and see what happens to the interaction with the plant.
Okay. Thank you, Professor.
Thank you. Thank you.
Thank you, sir. It was excellent discussion and excellent talk. And now I request Dr. Robin Gogoi, sir, if there are any more questions. If not, now I request Dr. Robin Gogoi, sir, to express vote of thanks.
Okay, thank you, Dr. Prashant. So here I am very much delighted to hear and discussion from Dr. Benjamin Horwitz, and I came to know his name is Benny, also popularly known as Benny. So we are very much thankful to you, sir. So you accepted our request to give your best presentation. So you have enlightened this works. I'm sure that this is only a part of your work. It is not as a total, because you are really a very sincere and in-depth worker. So that's why in this Platinum Jubilee lecture series, so you have given this talk and added value to our society. That's why, so on behalf of the society as a secretary, so I offer my heartfelt thanks, and also all the audience and members, students who participated. So this nice lecture. So on behalf of this Indian Phytopathology Society, I offer thanks to all of you. So I hope so you will continue to join in our other lecture series. So today is each lecture of our entire series. So this series will be continuing up to March. till our this international conference will be held in the last part of March 2022. So, Dr. Benny, thank you once again.
Thank you.
Thanks, Dr. Benjamin.
Thank you, Dr. Pratibha Sharma ma'am. Would you like to say some words?
Yes, yeah.
Thanks a lot, Dr. Benjamin. We are really glad to, it was really wonderful presentation, and I think all our trichoderma workers have really joined on this team. So we expected a lecture on that also, but nevertheless, this lecture was very, very important for all of us, especially the people who are working on the cell signaling and this. Dr. Shanmugam, actually, I was thinking he should ask the question, and he was there actually. So we are really thankful to you, Dr. Benjamin, and we look forward to your own, maybe you can also join us in the international conference if, we are trying to make it in a hybrid mode, and if it is possible that you come to India, I think we really cordially welcome you to come to India. Thank you very much again. Thanks. Yeah. Thanks to the whole team,
thank you.
to the whole IPS team. Thanks, Dr. Arup.
Thank you. Namaste.
Thank you.
Namaste.
Thank you, madam.
Thank you, sir.
Thank you so much. It was an honor to talk in this series.
Okay.
Hi, many thanks.
Bye-bye.